tenure in 2020, and since last year, I took up the position of the director of the Mississippi River Research Center, which is also part of the School of Agriculture and Applied Sciences. So with this background, I will jump right into this presentation. And my vision for this position is broadly based on these three pillars. First and foremost, I use the NMSU all-call-day strategic plan to guide my vision, and also the pertinent points in the strategic plan, most importantly soil health and climate resilient agriculture is going to be my focus, and most importantly the research and the outreach that is going to be conducted here is based on stakeholders and the farmers and the ranchers and whoever this station serves. So this is my three broad pillars, and I will touch upon all of them very quickly. The first one is the MSU strategic plan. I'm sure all of you are familiar with this plan. What caught my attention in this plan is the second sentence in the mission, which says, through testing of different crops, varieties, and production techniques, the goal is to provide new information that producers can adapt to their own operations for greater productivity and profitability. That is something that I relate very well to, because this is something I routinely participate in my current position. And then the opportunities are very nicely laid out. First is soil health, regenerative agriculture, and the second one is water. Third one being value-added crops. So again, I can relate to this very well, and I will give you some examples of how I have approached these topics in my present position. Going further into the strategic plan, it's very nicely laid out, and the recurrent theme in this strategic plan, which I found very interesting, is of course the regenerative agricultural practices that is connected to building soil health. And you can see where all the arrows are pointing. These are the key themes that are found in the strategic plan: regenerative agriculture, soil health, and if you look at objective 2.4, it's all about microbial community diversity, ecosystem functions, nutrient cycling, and of course all of this is finally connected to how we share this information for our small farmers, ranchers, and the stakeholders that the station serves. So with this in mind, I want to give you some examples of what I have conducted so far pertinent to the strategic plan. I would like to give you some examples of the soil health research that I've conducted purely from a stakeholder point of view, and one purely from a research standpoint connected to the agriculture experiment station at Auburn State. So one of the projects that I would like to highlight is connected to my center, the Mississippi River Research Center, which was established in the 1990s, mid-1990s. And the mission of this unit is to advance knowledge of preservation, conservation, and improvement of water quality while balancing the economic and environmental sustainability. To serve one of the short-term goals of this center, which is to develop science-based information, analytical tools, and decision support systems to enhance conservation of agricultural ecosystems. I have a project on which I'm a co-PI, and this was a USDA and RCS cooperative agreement grant, which is titled Nutrient and Water Budget Management Using Novel Sensor Technology in Mississippi. This is purely for the benefit of the stakeholders to basically mine information from the practices that they already have in place. So very quickly I will give you an idea of the research design. So this is where the institution is located in Claiborne, and that is where the Mississippi River Research Center is located. And so in this project, we identified 8 different farmers who have conventional practices and conservation practices. scattered across the state of Mississippi. And these 8 farmers have partnered with us to allow us to use sensors, the probes, which is again in a collaboration with a company. And then the probes have been already laid in these different farms. And this is the field design which I came up with for these farms, where we collect data before planting and after planting, and a multi-year study. So this is a 3-year study. So we're not telling the farmers what to grow or what not to grow. They do whatever is your traditional practices, but we mine the soil health information from their practices, so we can build a story about baselines. We can build a story about how their practices is related to the nutrients and the water management in these sites. So I was tasked with developing a farmer survey before we began this project, so I'm very familiar with developing farmer surveys and getting IRB approvals and all of that. So the first step and the second step, the farmer survey and the design, I developed that. And then validation of the probe information is also one of my tasks, which is getting the traditional information of soil health and then validating the probe information with whatever is the conventional way of getting soil health metrics. So this is an ongoing project, and Dr. Ayers Elliott, she is an urban farmer. She has, I think, about 15 acres of land in the middle of Jackson, which is the capital of Mississippi, and she was recently awarded the Woman Entrepreneur of the Year, and she has Footprint Farms, and she's one of our collaborators. So we have our probes also installed in her field, and you can see the probe that she's holding. So we've installed multiple such probes, and this year we got newer probes, which is sending information to our base station. So this is an ongoing project. This is just to give you an example of what I'm doing with the farmers and stakeholders. Another example of working with the stakeholders is also in conjunction with the Mississippi River Research Center is to identify and evaluate nutrient management strategies best suited for small-scale, low-income agricultural production. So these are the 17 counties that the university serves, nicely colored with purple because that's our university color. And you can see the institution is located right here, and this is the Mississippi River Healthy Watershed. So this is a new project that was funded last year. I'm the primary investigator of this project. This is Climate Smart Agriculture for Mississippi Small and Disadvantaged Farmers and Landowners. So this is a multi-state project. We have 19 different 1890 institutions. So there are 19 of them, and all 19 of them partnered together to develop this mother proposal for climate smart strategies for the small and disadvantaged farmers, because all of the 1890 institutions serve the small farmers. So this was tailor-made and specifically written for the purpose of the state of Mississippi. So in this project, which was funded last year from USDA Evans-Allen, there were 3 main objectives, basically to assess the stakeholder perception of climate change. So again, I started developed surveys for this, because even before we can offer services that are tailor-made to our clientele, we need to know what their perception is. Are they ready for climate smart technology? Are they ready for climate resilient agriculture? So that is our first objective. The second one is, of course, to collect agronomic management data from our stakeholders on their soil health and plant health parameters. And of course, the third one is to develop partnerships with our local USDA and RCS, USDA ARS. We have the Southern ARS in the state of Mississippi. So this is broadly our objectives, and of course, they have their sub-objectives. But the primary reason here is to be ready, to be armed with knowledge that can help our farmers for being climate resilient in the coming decades. And if you're familiar with the information from NOAA, which is the National Oceanic and Atmospheric Administration, they provided the state summaries last year. And for the state of Mississippi, we are expected to have warming, which is universal. But in our state, warming is not as much as a problem because our temperatures increased only by 0.1 degree Fahrenheit. But the problem for us is flooding and also coastal level going up. So the coastal levels rising and flooding and the vagaries in soil moisture is what we're dealing with and what is predicted for the next century. So our objectives are tailored to meet our state NOAA summaries. So this is, again, an ongoing project, and I have already completed my surveys and I'm awaiting IRB approvals. So that is as far as stakeholders are concerned. I will quickly tell you about a research project that I completed last year, and this was a long-term project on conservation agriculture in our experiment station. We have a Center for Conservation Research, which was established in 1988, and Dr. Paneker is the director of that unit. And in 2012, he started a switchgrass conservation study to understand how 4 different lowland cultivars of switchgrass and 2 planting densities, how they can improve soil health. And so this is completely cultivated under conservation ag, or otherwise known as regenerative agriculture. And I used this established site to develop a USDA Agri grant, which was in 2017. It was funded in 2017. And this was, again, a very ambitious project to study how the soil plant microbiomes change over a growing season over 2 years. So you can imagine the amount of data that you can collect from this. Microbiome was strictly bacterial microbiome and fungal microbiome, and how they changed in the soil, in the roots, and in the leaves to see if the microbiome actually was coming from the soil, because there is a prevalent theory that the soil microbiome is the reservoir from which the microbiome of the plant diverges. So that was the main objective, but in addition to the microbiome work, the goal was also to obtain the soil health factors. So we collected this at 6 time points. You can see the 6 time points. It's week 1, week 2, 4, 8, 16, and instead of having 32, we had it as 38. So you can see the progression. It's easier for our statistics, downstream statistical analysis. We chose week 38 instead of 32 because week 38 is when the frost hits, and then you have the above ground biomass has completely undergone senescence. So this is our time frame. We did the collection of all of these different samples at these 6 time points. This became a humongous project, and this was developed in collaboration with Kansas State and USDA ARS at Stoneville. Again, this is a purely research project. So this project trained and mentored 6 undergraduate students, and 2 master's theses came out of this project. And right now, this master's student who defended recently is already employed with USDA and RCS as an urban conservationist in Chicago. Anna is, she started off as an undergraduate at Alcorn, and then she went to Kansas State at RH Opponent's lab. She pursued a master's with the same project. She transitioned there because of the collaboration, and then she's right now pursuing her PhD program, and that is Dr. Heather Tyler from USDA ARS, where we did all of the enzymatic analysis. So this is a very nice collaboration. So this gave us tons and tons of data, so I won't overwhelm you with all the data. I'll just quickly tell you the synopsis of what happens. You can see all of these different parameters that were measured, soil chemistry. Then for the soil biology, in addition to the microbiome work, we did the PLFAs and the extracellular enzyme assays. And overall what we found was that the abiotic and biotic soil characteristics were very minimally affected by the cultivars and planting densities, but they were very strongly impacted by temporal changes. So in the beginning of the season, when the plants are still growing and emerging, you had these changes going on, and it kind of tapered off towards the end of the season because we're dealing with a perennial grass here, and obviously agronomic practices did not impact it, or it impacted it much less than the temporal impact. And very quickly, if you're interested in knowing about the microbiome information, this is what we found, both for bacterial and for fungal microbiome. Overall, we found the same trend. The temporal dynamics masked the minor impact of management choices, which is the 4 cultivars and the planting densities. So this is again only from 2018. I have 2019 data set as well, and I won't go into those details, but this is just to give you an example of how I've done pure research as far as soil health is concerned, and also stakeholder-driven research. Another example I want to give you about how I've engaged with stakeholder needs is my project on the specialty crop. Because I'm primarily a fungal ecologist by training, so I've also engaged in growing mushrooms, fungi. This is a very interesting project that I undertook as soon as I joined Alcorn State. And primarily I was tasked with studying shiitake mushroom for year-round cultivation and indoor cultivation. And the primary motivation for that was to augment farmers' income, all right? Why shiitake? Simply because the state of Mississippi is very well endowed with forestry resources. Shiitake is a specialty mushroom, and it can grow only on sawdust or wood. So we have tons and tons of sawdust, actually to be precise, 64.5 million cubic feet of sawdust that's produced annually, and 95% of it is just burnt for fuel. So this was used to basically value add for the sawdust industry. So Mississippi is not considered a mushroom production state in the USDA NASS statistics, so we thought that this was a great niche to fill, and there was already a established cooperative that Alcorn was funding and a part of, and it was known as the MNPA, which is the Mississippi Natural Product Association, which was established at New Hebron in Mississippi, and that was, I think, in operation for a good 14 years, if I'm not mistaken. And New Hebron facility actually started off in a garment manufacturing plant, which was abandoned. So that was converted into this shiitake mushroom facility. It was a very well-run facility where the blocks were produced, and the blocks were then sold to the farmers, and the farmers would maintain them under the required conditions, and they would come back with the shiitake mushroom. They would sell it back to the co-op. So my interaction with MNPA early on in 2014 was to help them reduce the cost of each block so that we could have more yield and less input costs. So that way we could help our farmers by changing the combination of the substrate block. And this is the research that I undertook in my experiment station to play around with the substrate composition to see if we could have less expensive inputs per block. And for that I scored a specialty crop block grant. And moving on from the crop block grant, and also because MNPA was shut down in 2017, I transitioned to a McIntyre-Stevens USDA forestry grant, for which I serve as the primary investigator. So I wanted to fill that niche and develop a similar facility on campus, and right now we are building this as we speak, and I will quickly show you what we're doing. So you can see this is the architect's plan. This is for the Alcorn State University Mushroom Facility on-campus research and demonstration unit. We already have an existing building which is undergoing renovation. And this is the plan. Let me give you a quick idea of how this plan is. This is a 3,000 square feet facility that we will have on campus in our experiment station to grow mushrooms indoor year-round. And it's going to serve as a research and demonstration unit. This is funded by USDA McIntyre Steenis. And eventually what is going to happen is it's going to look very similar to this, where we have these blocks that will be produced. And initially we want to have 50 bags, and then long-term is 200 bags per cycle. And you can imagine how big this project can end up being, because you can get easily 4 pounds of mushrooms from each block, and they sell at $4.50 per pound. So, and we also have a product development center on campus. So the goal is to channel everything from the unit here for obviously to sell fresh shiitake, dried shiitake, and also have a cottage industry where they can even make soaps and other sustainable products. So that is the long-term pipeline, but the pipeline is already there. And then of course this is also going to serve as a unit for teaching, research, and demonstration for our students as well. And then we would also like to provide technical assistance to our farmers, and more importantly, provide the spawn for our farmers, so they don't have to go looking for it anywhere. We just give it to them ready-made. And so this is what I'm working with right now. So without, this is my stakeholder engagement examples. So with these, with this in mind, I want to tell you very quickly what my vision going forward would be. And again, my guiding principles is coming from the strategic plan, where the key terms have been soil health, soil microbiology, nutrient cycling, and ecosystem functioning. And for that, the first thing from the point of view of soil health is, we all know that there are hundreds of metrics that can be measured from anybody's soil. Any agronomic system, you can measure more than 35 different response variables. Any classic textbook will tell you that. But what is that is most pertinent to our farmers, landowners, or ranchers? And for that, again, we don't have to go too far. Last year, a publication came out from the Soil Health Institute. And what they did in this ambitious project was, this was a multi-million-dollar project that was undertaken over several years, had a participation of over 100 scientists from 124 experiments. You can see the map. They were the sites of all these different experiments, and it had 6 different agronomic practices where you have residue retention, rotation diversity, crop count, cover crops, organic nutrients, and decreased tillage. So these were the 6 management practices that were studied. They evaluated all of these response variables and finally found that all of them could be distilled into just 4 parameters that are useful for measurement as far as our farmers are concerned. So if a farmer comes to us and asks us, what is it that I need to measure, instead of giving a long bucket list of measure X, Y, Z, it is now only 4 of them. And what are those 4? It is organic carbon concentration, carbon mineralization potential, aggregate stability, and water potential. That's all. So 4 of them that make the most sense. And also this beautiful study actually showed how cost effective it is and how you can get all of this in any general soil testing lab to give you a metric of soil health. If we want to measure additional parameters, that's fine. That's purely from a research standpoint. But for practical considerations for a farmer or a landowner to make any kind of assessment of their land, it has now been distilled into these 4 parameters, and this was published last year. So I do follow up with these presentations and publications that Soil Health Institute has, because they have given some really good guidance after this big project that they undertook. And in fact, 2 of my students interned in the Soil Health Institute, and one of them undergraduate is now going to Cornell University to pursue a horticulture program. Another important thing I consider valuable in this position is to actually look at not just the soil health, but I like this guidance from the Soil Health Institute, which came from their annual meeting last year, that is to understand the metrics at business as usual. Farmers have set practices, so business as usual, what is the metric? Then find out the metrics when you have sustainable practices, which is the SHMS, the Soil Health Management System, and then come up with a target. What are the practices that can push us to that target? I think that is even more valuable. So we have soil health targets. These are the 4 parameters that I previously talked about, and I think this is very, very valuable going forward. This is an incremental improvement in soil health assessment. So not just measuring something, but more importantly, are we moving towards, or what are those practices that are helping us move towards that target as far as soil is concerned? The next guiding principle for me, as I mentioned previously, is the guidance coming from the National Oceanic and Atmospheric Administration. They gave out the climate summaries last year, and if you look at the New Mexico summary, 3 key messages. Unlike Mississippi, which saw only 0.1 degree Fahrenheit increase in temperature, warming is not a huge problem in Mississippi, but it is here in New Mexico, which is temperatures have risen more than 2 degrees Fahrenheit. So warming temperatures are really something to be worried about in the next century. So are we prepared for that? What are the economic practices that will help us prepare to face this challenge of unprecedented warming? The second one is drought in the state of Mississippi, in the state of New Mexico, where the intensity is again projected to increase along with wildfires. Then the third one is uncertain precipitation. Again, adds to the whole drought story. So these are the 3 key messages for the state of New Mexico. So using this guidance, how are we going to prepare ourselves in the future and give the required guidance to our stakeholders? Another guiding principle comes from the USDA Sustainable Practices Goal that was put out last year, which is the reduction of greenhouse gases coming from agriculture, to try to go towards a carbon neutral or a greenhouse gas neutral agricultural system. 11% of our greenhouse gases are released from agriculture, and these are some of the most common practices that release the greenhouse gases. And so how can we have these practices but still make changes in slight changes in our agronomic practices to reduce greenhouse gas emissions? Because all of this is eventually tied to the climate smart and climate resilient agriculture. So with these guiding principles, what is it that we can do here? Well, of course, you have a very well-established site where you have all these different lots set up. You have long-term fruit crops and all of that going on, certified organic farming, so fantastic. So the groundwork is already laid. But I think with all of these guiding principles, it's very important that we investigate new varieties that are tolerant or resistant to the climate challenges that we talk about. New varieties of crops that are drought and heat tolerant and also tolerant to pathogens. Because one other consequence of increasing temperature and precipitation is microbes turning pathogenic. Previously, the ones that were not pathogenic turning into pathogens. Now there are enough studies that have shown that. So trying out new varieties already, I think, is a very important point so that we are climate ready. And I have already undertaken projects of this sort. I'm also investigating the microbiome of Freedom giant miscanthus, which was specifically released as a drought and heat tolerant bioenergy grass for southern United States, southeastern United States. It was released by Mississippi State, but now it's a commercial variety. And I have also looked into the long-term soil health of growing Freedom giant miscanthus. My student is working on it as we speak. So very familiar with the whole drought and heat-tolerant variety trials. Of course, we can't take these studies at one time in a year, one-time point. This has to be ongoing. And the most recent guidance coming from the microbiome world is don't look at who is there and what they're doing at 1 or 2 time points. See how they're changing over a period of time. Shifts are more important than just who is there at what time. So look for indicator microorganisms and how they're shifting so we can better predict our agro-ecosystem. And it's very important to do community profiling, which again nicely ties with the strategic plan. And what I mean by community profiling is to look at them from a point of view of the biogeochemical cycles, the nitrogen cycle, the carbon cycling, and all of that. All of them tied together. And coming to the greenhouse gas emissions, this is something that I am looking right now to purchase from my Climate Smart grant. This is it's a handheld greenhouse gas emission measurement meter. It's like a backpack you put it on. It's produced somewhere in Europe. I think it's from Sweden, and it costs a good amount of money. But I think I want to get this and play with this toy next. It can measure all the 5 gases. So I think it's very important to invest in next generation equipment that can help us make these measurements. So and also once we get these, you can also measure the same from water systems as well, because they're not limited just to the soil. I want to give you quickly my last slide on community profiling and what I meant by community profiling. Last year, I got competitive funding from USDA to buy this baby. It's $150,000 equipment, but only this is $137,000. The other unit, I haven't shown it to you here, but the OmniLog helps with community profiling. It's a high throughput system. It's like a mini refrigerator. You open it, you have these racks where you can put these 96 well plates. All you have to do is take your sample, whether it came from a soil sample, a water sample, it could be a sludge digester, it doesn't matter where that sample came from. It has to just be plated on this pre-made 96 well plate. You have these different types of plates with carbon, nitrogen, phosphorus, and all of that. And you can make your own plates with pesticides and chemicals and see how the degradation pattern shows up. And the pattern will light up like this with the purple dots. And that's what this unit is looking for. So, of course, it does phenotypic microarrays. That is purely from a research standpoint. Can be a nice master's or a PhD project. But for us, how can we use this in conjunction with the farmers or ranchers is they can bring us the samples. If we have this unit and we have these plates, you can get the information in 24 to 48 hours. What does it tell? Yes, it's a bunch of dots, but the dots are telling you who's there and what they're doing. It's not individual microbes, but we're looking at the entire community. So I already have this. I acquired this equipment as a part of classroom instruction for the different undergraduate courses that we have, but it is easily usable for undergraduate research, graduate research as well. And we also hope to use this in our field days to demonstrate it to our farmers. So this is something that came last year. So eagerly looking forward to testing it with other water samples. With that, I will stop, and I think I have addressed my vision here. And if you have any questions, I'll be more than happy to answer them for you.